Hello and welcome to the Sentinel Innovation Center webinar. I'm Leslie Curtis from the Duke University School of Medicine, and I'll be moderating today's session. The U.S. Food and Drug Administration's Sentinel system helps to address questions related to the safety and effectiveness of marketed medical products, and the Sentinel Innovation Center focuses on developing innovative methods to further advance Sentinel by leveraging new data sources, building new tools, and enhancing real-world evidence capabilities through advanced analytics. We launched the webinar series to engage experts and innovators in key areas related to the Innovation Center's master plan. And with that, I'm pleased to introduce today's speaker, Dr. Keith Marsolo. Dr. Marsolo is Associate Professor in the Department of Population Health Sciences at the Duke University School of Medicine and a member of the Duke Clinical Research Institute. His research focuses on infrastructure to support the use of electronic health records and other real-world data sources in observational and comparative effectiveness research, as well as standards and architectures for multi-center learning health systems. Dr. Marsolo is co-investigator in the Distributed Research Network Operations Center of the Coordinating Center for the National Patient-Centered Outcomes, Patient-Centered Clinical Research Network, or PCORnet, as we shorten that frequently, where he serves as a faculty lead for activities related to the PCORnet Common Data Model and data curation. He's co-lead of the PCORnet Data Work Group as well as the Common Linkage Work Group. And he also serves as a member of the Data Quality Subcommittee of the National Evaluation System for Health Technology Coordinating Center, or the NEST-CC. The title of Dr. Marsolo's presentation is Data Curation in PCORnet: Lessons Learned and Implications for Regulatory Decision-Making. Now, before we get started, I'd like to mention that your lines are muted. If you have a question, please pose it by typing it into the Zoom question and answer feature. We'll answer questions at the end of the session. As you probably heard when you joined, this session is being recorded and will be made available through the Sentinel webpage, which can be found at sentinelinitiative.org. So without further ado, Keith, thanks so much for joining us, and I'll turn it over to you.
Great. Thanks, Leslie. Thanks for that introduction, and thanks for having me on the webinar series. So yes, today we'll talk about data curation in PCORnet and lessons learned, and then what are some of the longer-term implications. Just a couple of disclosures. I think first, probably the main one is just Duke is being funded by PCORI to support the PCORnet Coordinating Center, and the statements here are really my own and do not necessarily represent the views of PCORI or PCORnet. So what I wanted to do today in terms of goals is just describe some of the current practices. Again, the lessons learned from efforts in PCORnet to assess data quality and dataset suitability, and then the implications for using EHR data more broadly to support regulatory decision-making. So just a little bit of background around PCORnet before we dive in, just to level set everyone. I think if people are familiar with Sentinel, most of this should look familiar, but again, just wanted to make it clear so that we're all on the same page starting off. So PCORnet is a network of networks, and so it's made up of clinical research networks, which would be networks of academic health centers, health systems, things of that nature. There's networks that are comprised of federally qualified health centers. There's also health plan research networks, and so those are more familiar for those that deal with Sentinel. So claims-based partners. We have patient partners, and then a coordinating center. And so the coordinating center is made up of a number of institutions. The data and query-related functions of the coordinating center are split between the DCRI and then Harvard Pilgrim Healthcare. And so all of that together makes up PCORnet. In terms of the way that PCORnet operates, its query platform or its data platform, again, it's a distributed network where essentially requesters come to the front door of PCORnet. That with a question. That question gets translated into a query package. The query package is sent to partners, and then the results are aggregated back at the coordinating center. And so following the traditional distributed network process. This map illustrates where the PCORnet participating sites are located across the country. And so the pins are essentially the lead sites of each of the networks, and then the dots represent, again, just participating members. And so it's roughly 70 partners across the entire network when you count them all up. The PCORnet common data model. So this is a cartoon that illustrates many of the domains within the data model. This isn't everything, but we just wanted to highlight a few. So the ones on the left in the green are the ones that we would say were more ready for research or more well curated, have been used more for existing studies. And then the ones in the blue on the right are available, but they're more at the pilot stage, and so you can get a sense of what's in the data model. The PCORnet data model was originally it was evolved from the Sentinel Common Data Model, and then a number of extensions and modifications were made in order to better support the EHR data that existed at the various partners. So when we talk about data curation in PCORnet, the process that we use is essentially, it's a two-stage process. And so we have what we call foundational curation, which is really to establish a baseline level of data quality. And then there's study-specific curation or study-specific characterization. And that really is a deeper dive to make sure that data are fit for purpose for a given study or analysis. And the cartoon on the right there is meant to illustrate that process, and it's taken from a white paper that came out of the Duke Margolis Center around characterizing real-world data, and so it's something that we like to use to describe our process. But I think one of the things that's important to note around data curation in PCORnet is it's not static. It is a continuous process, and so there's a continuous assessment of performance, and so it's something that we run essentially every time partners refresh their data, which is once a quarter. But then the process itself is constantly evolving, where we're looking to close the gap between foundational and study-specific checks, where we add new checks based on study findings or on the results of queries. And so we've got the PCORnet sort of definition or framing of assessing fitness for use, but I thought it would be important to talk about how does that relate to the FDA definition of fitness for use, because it's not exactly the same. So I thought I would just point that out here. So this language is taken from a guidance document that the FDA released, the use of real-world evidence to support regulatory decision-making for medical devices, and then it was carried into the real-world evidence framework that was recently released. And so when we talk about fit for purpose in the FDA document, it's really around assessing the relevance and the reliability of the source and its specific elements. And so when we talk about relevance, you can see the factors that go into relevance here, but do the data apply to the question at hand? If you're going to use them for analysis, do they let you answer the question that you're trying to answer? And then, the results that you get, can you actually interpret those? Do they make sense? And so those are some of the factors that go into relevance. And then when we talk about reliability, that's broken down into two factors of accrual, which has to relate to how the data are collected. And so, are there manuals that describe operations, elements, data element definitions, methods of aggregation, things like that. And then assurance, which is really around sort of the quality control standards to ensure that data and analyses are reliable and trustworthy. And so the example there is like some of the best practices that came out of a number of the patient and device registries. And so, real-world data sources are a little bit different in sort of how they evolve, how data are populated, and so they're not necessarily expected to fill all the characteristics of reliability. But I think a lot of the principles still apply, and we'll talk about some of that as we go. And so if you map sort of the PCORnet process to the FDA definition, sort of how do those things correlate? And so what I would say is that the foundational curation process, which is what I'm going to mostly talk about here, is focused really on more on the level of data assurance, although there are some aspects of accrual and then relevance that are that apply, but mostly on assurance. And then when we talk about study-specific characterization, that really gets into relevance because you're directly looking at the population and data elements of interest. And so you might ask, why is foundational curation focused on data assurance? And I think the main reason is that for many EHR data domains, they're really being harmonized and standardized for the first time. And so this is maybe a little bit different from administrative claims, where they're kind of a known animal, and there's been years and years of research on sort of large scale surveillance and research on those data, and so it's much more of a known entity. EHR, it's just not that, we're not at that point. And so we're harmonizing, we're standardizing really for the first time at any kind of scale. And given the volume of data, it's overwhelming to do that harmonization while also assessing fitness for specific study questions. And again, just to illustrate the point, the graph that's on the shown on the slide here, it's essentially the growth of lab results within PCORnet over the last 4 years or so. And so what you can see is the bars represent the number of lab results that were available in the data models of PCORnet partners in billions. And so 1.5 in 2016, and then 11.5 after our latest refresh in July of this year. And so that's a huge number of just results. And then you can actually see the line is the number of the median number of LOINC codes or tests that are available in the common data models of the different partners. And so when we started, we were using a relatively constrained list of lab results. And so it was just a handful per partner. And then now over 1,800 lab tests at each partner in their data model. And so it's just a huge number of data. And I'll give a couple other examples here. So this is an example of the encounter types that are available within just one EHR. And so EHRs are going to have dozens and dozens. I think this is around 170 different encounter types. And so there's an exercise that has to be done to map those encounter types to the value set that you would see within the common data model. And so the PCORnet common data model value set for encounter type is shown there on the right. And so and this is actually something that we're going to have to address here in the next few months, because you may notice that telehealth is not actually called out as a specific encounter type, but given the COVID pandemic, and the explosion of telehealth visits, that's something that we want to be able to call out explicitly. And so that's another thing that we're just going to have to extend. For lab results, this is I talked a little bit about labs, but this is actually the sort of the process that somebody would need to go through in terms of harmonizing their lab results. So this is if they were looking at hemoglobin and they wanted to choose the appropriate LOINC code for hemoglobin, they have to know a number of different things around the test. So the name of the test obviously is important, but then sort of the property, whether it's mass per volume or just looking at presence or moles per volume, that's important to know, and that's based on the unit of measure. The specimen source is important, and then in some cases the method itself, the instrument that's used. And depending on who's doing the harmonization, they may not be familiar with all of these concepts. They may not have access to all of the data that they need in order, to fully specify all of these things. And so it's just a process that everybody needs to go through. So when we talk about, foundational curation and foundational data checks, what do those tend to look like. And so when we design them in PCORnet, they're really focused on the categories here. Do the records conform to the structure and the format of the common data model? Are records internally consistent? And so if there's a LOINC code that's specified for a lab test, does the specimen source match the LOINC code? So if the LOINC expects a blood sample and somebody says that the specimen source of the record is urine, that's a potential mismatch. If data are going to be used in an analysis, are all the necessary fields populated? And this one can cause trouble in the EHR when some fields are less well populated than others, particularly for some historical data. And then, do the values make sense? Or are they plausible? And so those are the things that go sort of into the design process for foundational checks. But then the things that we have to keep in mind is, some fraction of the data will always be dirty. No errors is a problem. We don't want to see things too clean. EHRs are really, they're constantly evolving. But in particular, older data, particularly before 2014 and some of the meaningful use regulations, are a lot less standardized than the data are today. And then, you also need to allow for variation in population or practice patterns. And so this comes into play when you're setting your failure thresholds. And so, what might be considered the normal range for blood pressure, for instance, means one thing in an adult population than it does in pediatrics. And so if you've got a network with a wide variety of patient populations like you do in PCORnet, you'd have to sort of allow for that. And so all of these things can help determine what checks are required and what are optional. And this is something that we struggle with a lot when it comes to PCORnet, because again, as I just mentioned, when you have a wide variety of populations, you either have to set very broad acceptance criteria to the point where it's meaningless, or some of these things are just more optional and informative. So the data checks within PCORnet, they tend to fall into the categories that are outlined here. So conformance, I mentioned, the data adhere to the format of the data model. Completeness, there's values where we expect them. Plausibility, where they make sense. And then persistence, where the patients and the records, essentially, they don't disappear between refreshes. And you can see the growth in the number of data checks over time. So those are represented in the bars, and then the various data models that were in place at that time. And then the line is essentially the number of what we call data check measures. And so that number correlates to, if there's a data check, for instance, fields do not contain values outside of the CDM specification. If there's 50 fields that have a value set within the data model, there's 50 things that we need to check. And so the number of checks can translate into a huge number of check measures, simply because of things like conformance, where you just have to check a number of different fields and tables in the data model. So I'll quickly show the different data checks in PCORnet. I'm not going to walk through all of them because I don't want to put everybody to sleep. But so the conformance, as I said, the bulk of the check measures fall into the conformance category. And the conformance ones are largely the checks that are required within the data model, because these are things where everybody should be able to meet these. And so you can see examples of those here. For plausibility, these are investigative checks, and some of this is because, again, it's the first time that it's being harmonized, and then others is sometimes there's sort of reasons why a site may or may not, or may be failing these checks. So you can see some of the plausibility checks here. And then in completeness, again, these are the checks that we have that really look for, if there's a record, are the data where we expect them. So there's a number of checks related to lab data and mapping data, are things, essentially mapped how we would like to see them. And then looking at whether the number of certain items are below threshold, and I'll get into these in a little bit. And then the persistence checks, looking at decreases in the number of patients or the number of records. And so, you can imagine why this would be important if you're running a study and doing periodic refreshes, and you see a large number of patients disappear from one query to the next. That's a potential problem. And again, I'll talk more about that here in a second. So when we talk about data check failures, there's failures that are essentially remediable and non-remediable. And so, the non-remediable ones are potentially limited to, or can be because of population characteristics. It's just the nature of, who's receiving care, or the characteristics of the health system. So, if there's a health system that doesn't have any inpatient data, any data check that as an inpatient flavor, the performance may be different for that partner. And then there's, source system limitations, either that the data doesn't exist or there's some sort of system artifact that, causes it to be populated in a way that's unexpected. And then there's, the remediable ones are, there's a problem mapping to the reference terminology or the value set. So from going from source to the target model, something was messed up there. There's different types of source system limitations where essentially the people that are populating the data mart don't have access to whatever clinical or billing system might have the data in question. And so we've seen that happen in cases where, again, people are on the research side of the health system and they have access to, certain parts of the EHR database and not others. And so those are source system limitations, but they're ones that we feel that, with enough effort, sites should be able to address those. And then there's issues that are introduced just by the way that partners are populating their common data model. And again, I'll talk about some examples there. And I think what we in PCORnet have to deal with is that it's hard to say, for any given check that everybody should definitely be able to fix whatever issue it is that's faced. I mean, there's some where we think that everybody can broadly improve, but there are others where performance may just be what it is, and then the purpose of the check is really to just inform analyses and then allow people to make a decision on whether the issue that's there is going, whether or not it's something that they can, design around or mitigate when it comes to doing their analysis. So I'll talk about a few of the results here for PCORnet. And so what we have here are some of the key foundational data checks. And so what you're looking at is the pink line is essentially the percentage of medication orders that are mapped to what we would call a preferred RX norm term type. And so there's a number of different sort of valid codes that for any given medication order that describe it at varying levels of granularity. And so the preferred codes in PCORnet are the most granular. They're the ones that encode the most information. And so there's a check that looks at whether sites have 80%, at least 80% of their medication orders mapped to that preferred term type. And so you can see that we've gone, starting at about 50% in 2016, and I think we're about at 80% for the whole network. And so I think that's around 3 billion medication orders, for instance. And so there's been some improvement there. The lab mapping this is the percentage that are mapped to LOINC, and so you can see that we've got a pretty good growth there. Again, for approaching 12 billion records across the network. And then the blue line is the latency of the data, and that just has to do with once the data are refreshed in the data model and they run curation, essentially how current or how recent are the data. And so what we try to say is that the data are at least at least 3 months or less in terms of latency. And you'll notice that that number really tanked in the last refresh, and actually that has to do with the COVID pandemic and the fact that the number of visits to most health systems decreased dramatically. So the way that latency is calculated is we look at the sort of, there's a baseline that's computed over the sort of 2 years prior to establish the average number of encounters per month or diagnoses per month or whatever it's events per month, and then we look at whether or not the most recent month or the most recent couple of months were within 75% of that total. And so when the number of encounters decreased dramatically, it looked like everybody failed, but it just has to do with the fact that essentially the system changed, and so we can't necessarily use previous benchmarks. And so that's the other thing. I think it's actually been a good lesson when we're designing these data quality checks, is that you can have things that drastically change your system, and then people all of a sudden start to fail, but they're not actually failing. It's just an artifact of the system. And so this graph is meant to show some selected completeness measures, and I think the takeaway here. So this is looking at the blue line is looking at future dates, and so essentially the percentage of the network that have fewer than 5% future dates in their data model. So things like you don't have events that are in the future, diagnoses in the future, things like that. Again, there's always a certain percentage that are expected, but people should do pretty good. Primary diagnosis, just looking at the number of sort of inpatient diagnoses that have a primary diagnosis flag encoded, and then looking at illogical dates, which is the orange line. And for the blue and the green, again, these are relatively stable performance, and so this is just an indicator of something where the sites that are able to fix it have fixed it, and then it just exists. And so, like for primary diagnosis, it's one of those things where if you've got an analysis that relies on primary diagnosis, so many claims algorithms based on claims data will take primary diagnosis into account, and so if it's something that is in your study design, and you're choosing to rely on primary diagnosis, it's you're either going to limit the partners that you can use in your study, or you've got to think of a different approach. Now, the illogical date relationships I included in here, not necessarily to highlight the fact that we have a bunch of illogical dates in our data model. It's actually that we changed the definition of the data check and to partway through to actually look at looking at whether we saw sort of procedure dates that were before an admission or after a discharge, for instance. And what we actually found is that there was a number of sites that were chipping that check, and it just has to do with idiosyncrasies between sort of procedures as they're captured in the billing system, and then sort of dates in the EHR. And so it's just one of those things where technically it's an illogical date, but there's a rationale behind it, and it's just one of those things that we have and we report out so that people can include it in the study design. So that's just one of those unintended consequences, but I think it's informative of what can happen. And this camouflage slide is meant to show the data persistence checks that we see within PCORnet. And so what this is every row, what you're seeing here is a data mart in PCORnet, and then the columns are refreshes. And so there's the three big columns correspond to the 3 persistence checks. And so what you're seeing is performance over around seven refreshes for the network for the 3 checks. And the green indicates that they passed the persistence check, and the red that they failed, and then the gray is essentially there was no refresh, or they weren't approved. And what you can see here in the red in January 2019 indicates sort of when we officially turned on the check. And so what this is meant to show is that, that the persistence was actually a fairly big problem where we were seeing drops in data or records. And it improved a little bit once we actually turned on the check. But we still see every time we refresh the data, and I didn't include more recent results again just because there's some weirdness with COVID. But we still continue to see these persistence issues, and in some cases, there are valid reasons for doing this, where somebody may drop some historical data. They may go through a process where they're sort of adding hospitals and deduplicating records, cleaning up code sets. And so there's reasons why this can occur. It's not something that we want to see every single time, because that kind of instability is not great. But it does happen. And so this is, again, one of those things where, we try to monitor it, and then we can use it to inform our study designs. So I'll talk next around, what are some of the things that we've kind of learned through curation, and how has curation influenced the common data model, and then what we've learned in studies, how does that influence what we do with curation. So I think when we were running curation, we surfaced a number of instances where there was just ambiguity in the specification of the data model. And so, we're continually surprised by, what comes out of the data. The data model right now is 200 and some pages if you print it all out, and we think it's pretty specific, but, it continues to surprise us where there's questions of edge cases when people ask us what to do. So, there's a death table in the data model, and we have partners who don't have any data depth. And so if date of death is a required field in the data model, they may need some guidance on sort of how to do that if we want to include those records, or if there's complexity in the source data. So if race and ethnicity are captured as a single field within the EMR, how should we break that apart to populate the relevant fields? And so what we ended up doing is we developed implementation guidance, and so that's additional information that we provide to the partners as they load the data. If they're faced with choice A or choice B, we want to tell people to choice A is preferred to try to reduce some of the variability that exists. And so, we've got general guidance that sort of touches on issues that affect the whole data model. There's table level guidance that is, related to a specific domain, and then there's field level guidance that we have for specific data elements. And you can see examples of table guidance and field level guidance here on the slide. So when we talk about studies, so we had some demonstration studies in PCORnet when we were just getting started that they were looking at prescribing data really before we had curated it as a network. And so what you can see here on the left is a screenshot from RxNorm, and for a record of acetaminophen 325 milligrams with hydrocodone 10 milligrams, what you can see here is these are all the valid RxNorm codes that you could assign to that record. And so the ones where it's like branded drug component, those encode a lot of information, or branded drug encodes a lot of information, or this clinical drug encodes a lot of information. And so those can be useful. However, these things here with like dose form group that tells you that it's a pill, if you had a code for this record and it just said pill, that's not very informative. And so, what we wanted to do was to try and create a hierarchy Rx norm term types, and then, and tell people to map to preferred ones. And so what you can see here is actually on the bottom right is each of the columns represents the data marked in PCORnet, and then the color coding is the percentage of records that were coded to the different Rx norm term types. And we created groupings for each of the color coding, where green is the most preferred, and the other colors are sort of less preferred. And you can see that there's a fair amount of variability that was that went into play. And so we created this table in the data model that basically rank ordered the different Rx norm term types, and again told people to on what to do. So we thought that was relatively relatively clear. However, as we further analyzed the medication data, we had to add some additional clarifications to the guidance. And so things like not assigning a CUI that essentially encodes more information than is supported by the source data. And so if we go back to this example here where, acetaminophen hydrocodone, if that's all is just the generic, you shouldn't be assigning a brand name CUI that a patient received a brand name if you don't actually know that. So there are cases where you can infer that from the EHR, but if you don't know what's being dispensed, sort of leave it at the level of the generic that was ordered. And so those are some of the things that we've had to face, and then also just dealing with the data as it exists at the source, and then making sure that that partners can actually encode the information, because we occasionally see weird things in the systems, and that it may not be clear on what they're supposed to do. So that's just an example of how we've learned from studies, and then we continue to update as we go. And then I think the last thing I'll in terms of study outputs is the data latency, and I touched on this earlier with the COVID example, but this is this is something that we designed to help some of the pragmatic trials that are ongoing within PCORnet. And so you can see here where there's events sort of in the red lines, and then if there's a query date here at the end, and then this big gap in the middle, does that mean that there were no events or there's no data? And so ignoring all of the questions around sort of complete capture of claims versus EHR, I think what we're focused on is if there were events that are in the EHR, do they make it in a data model, and then can we tell? Do we have confidence that we've captured the events that occur? And so that's what this latency check is supposed to look at. And so this is where looking at you look at a 2-year period, and so 24 to 13 months prior, you use it to compute an average number of events, and then with the most recent 12 months, you can look at essentially, what's the first month with any data, and then what's the first month with complete data, which would be 75% of the benchmark. And so that's what we can use to try and inform questions around sort of completeness, at least when it comes to EHR data. And then these are some pre-COVID examples that we have. The left shows essentially the average number of, or the number of months per each bar represents a data mart, the number of months latency per data mart. And so you can see most of the network is doing pretty well, and then there's some outliers there. And we try to work and understand what's going on. Is that something with the way that they're receiving their data from the source system? Are they refreshing the PCORnet common data model after they refresh 2 or 3 other common data models, so they're all in a chain? And so there's sort of artificial latency that's being introduced, or are there just sort of other things that are going on? And then I think the other thing to just note with this is that latency can fluctuate over time. And so this just shows 3 examples from the network where somebody's doing pretty well consistently, somebody was doing worse and has improved, and then someone else has performance that's going sort of up and down. And so again, each of these things can kind of tell you different things when you're looking at a data model for a study. So I'll briefly just close on some future work here. And so I think two topics, deeper assessments of source to data model mappings, and then looking to close the gap between foundational and study-specific curation. So with the source to data model mappings, there's some domains within the EHR just not captured in the terminology used for analysis. And I've touched on these: RX norms for medications and then LOINC for labs. The existing data checks that we have are really assessing whether the data model is internally consistent, and it's less capable of looking at whether the data model record is truly reflective of what's in the source. And so, given a medication, did you pick the right RX norm code in the first place? And so one of the ways we're thinking about doing this is looking at what are known as the raw fields that are in each of the data models that store information about a record as it existed in the source. And so these are often text strings that can be parsed to provide information. And this would be developing procedures to compare the raw and the encoded values to flag issues. And so an example here on the bottom, the right set of columns contain the medication string or the medication order as it exists in the source system, and then the left is sort of what was chosen from RX norm to represent that medication on the left. And so what you can see, where the ones that are highlighted, where there's a mismatch between essentially between the ingredients. And so you've got examples of multi-ingredient drugs that are encoded to a single ingredient RX norm code, or vice versa. And so, that's an issue if you're trying to design a study and you're using a multi-ingredient drug. You're going to miss, sort of all of those single ingredient examples. And so that's just something that's a big problem in an analysis. And so it's something that we will want to try to address going forward. And then when we talk about, closing the gap between foundational and study-specific curation, study-specific curation is really around, quality concerns with a variable within a given population. So this is an example that I like to use where you're looking at the percentage of a cohort with a lab result by data mart. And so essentially what each bar represents is the percentage of the patients in that cohort who have a lab result. And the ones on the right, the data marts on the right where there's no data, you can figure that they probably didn't load the data into the data model. But then the stuff that's kind of in between the 5 to 50% range, it's tough to know whether that, is that practice variation, or are there gaps in the data for, certain years or certain clinics. And so one of the things that we've done within PCORnet is we've tried to do these groups to look at, diagnoses, labs, and procedures, and then, whether or not we see them in the system. And so, it's relatively easy to know what to do if there's nothing, right? If we don't see mammograms in an adult hospital, that probably means that that's a problem, right? Those procedures are missing. But then, what percentage of patients with a mammogram, that can be very difficult to know what's a meaningful number against, an entire patient population. And so the kind of thing we're thinking about is these population reports, essentially, where we define a series of conditions, rare and common, and then for each of those, what are the comorbidities, the events, the medications, the labs, sort of the structured elements of interest, and then can we generate statistics across time and care settings, and then look at that to benchmark and try to determine what's outliers and give us a better sense of what's going on. So just closing out here, so I talked, some of what we've seen within PCORnet. However, I think maybe the one thing I'd want to stress is that this is inherent to sort of all EHR data. This is not PCORnet specific, so the problems that you see here are going to exist in other EHR-based data sources. And so, what we've tried to do to characterize that is really use data curation as a process for improvement. So both improving data quality where we can, but then also improving the quality of our methods so that we can start to look at other things. I think it's important that we continue to develop and share best practices around these fitness for use assessments. And then I think, the other key factor here is going to be, sort of how does that translate into the FDA guidance and sort of what the FDA wants to see in terms of fitness for use. And then maybe the final takeaway is, it's been years understanding the pitfall of working with claims data. And I think, we just have to have a little bit of patience to develop that knowledge as it pertains to the EHR. And so I think with that, should be able to have time for questions.
Great, thanks, Keith. That was really terrific. Just a reminder to everyone, if you have questions, please go ahead and type them in the Q&A box, and we'll take those and probably I'll take them in the order in which they come in. Keith, let me just start with a clarification question that came in. Can you describe what you mean when you talk about data curation cycles, or when you talked about when a check, what cycle a given check was added?
Yeah, and I apologize for that. That's where we've various versions of these slides. So when we talk about data curation in PCORnet, we tend to follow what we call our cycles. And so a cycle is generally 6 months where there's a refresh, sort of refreshes once a quarter. So 2 quarters represent a cycle. And the reason that we break it into cycles is because we try not to introduce new data checks sort of in between cycles. And so essentially what it'd be is we would do something in introduce new checks in January, there's a refresh in January, and then April, and then a new cycle would start again in July. It's not always been the case. Sometimes a cycle is 3 refreshes, but that's generally what we mean.
Great, thanks. So we've received a couple of questions about this concept of threshold levels for the data checks, and I would say the overarching question is, how exactly did we arrive at those threshold levels, which certainly, as one person notes, seem to represent a pretty low bar for something that we would call quality?
Right. Yeah, and it's something that we go back and forth with quite a bit. I think, again, knowing that there's going to be some level of quote unquote dirtiness in the data, because there may be valid reasons for it. So like the illogical date relationship one is it's a relatively easy one to trot out, where you would say, well, you can't have any procedures associated with a patient before they were born, and you can, because if there's sort of in the neonatal period, right, there's some kind of surgery or something, there's going to be some patients that have events. So there you'd say you'd want to set a small threshold, because 0 is actually going to throw out valid data. So that's sort of one thing. The other thing I would say is, some of this is thresholds that we would want to iterate or gradually increase over time. So you start at a fairly low bar, or low-ish bar, and then you can look to increase it over time. And so that's the other thing. And then maybe the final thing is, these are just basically the thresholds to, can you use these data to respond to basic research types of queries? And then for any given study, you absolutely may need to go with a higher threshold of the same kind of check. And so I think what we're trying to do is, what's kind of good enough for the basic work that we want to do. And then, we're examining sort of the types of things that we would want to examine for a given, study or analysis within a population. And then if you need to ramp up the threshold for that, that's totally acceptable.
Great, thanks. So you walked through some really great examples early on about some of the challenges in curating lab data that we've seen in PCORnet. And I'm thinking about the fact that we probably have many people dialing in today or listening today who are really familiar with the approaches that have been taken in Sentinel with respect to lab data. and which has been going on for quite some time. I wonder if you could touch a little bit on what maybe some of the key differences are between the kinds of the streams of data that we're talking about when we're talking about lab data that may be included or available from almost a claims perspective or an administrative perspective, and the kinds of data that we might be talking about in a native EHR. And kind of, does that matter, or how does it matter? So it's a big question, but it occurred to me that it might be helpful for people who are familiar with one but not the other.
Yeah, and you know, so I would certainly say that we've learned from the pain of Sentinel, and I think we've relearned some of that pain, and so certainly leveraging a lot of the work that happened there. I would say that the data are, in some sense, they're the same, but then in other cases, it's different in the sense that if you've got lab data from, like, going to an administrative claims partner, you know, that may be an interface where it's from an external lab, or, you know, or a reference lab or something like that. But there's just, it's sort of data that's thrown over the transom, if you will, from the lab to the claims partner. And there's not much of an ability to go back to the source and inquire around, you know, what are some of the, you know, whether things can be fixed, if there's a question on what's being received. And so I would say that's probably the main difference with PCORnet is you've got, you certainly get data that come from external labs, but then a lot of the lab information is also from the internal, you know, clinical labs to the hospitals and the health systems. And so there, you may have access to some of the lab personnel, or, you know, be able to go directly back to the lab information system or the instrument itself to get sort of additional information to clean things up. Now, I would say that, you know, the database folks on the research side may not be comfortable talking to those people, or may not know those people, and so there is, you know, an exercise of going through that to fix it. But it is something that we think should be fixable, because, you know, again, you're using these data to take care of patients, and the kinds of things that you want to know for research purposes, you know, what's the lab, what's the unit, what's the source, you know, that stuff's important when you're taking care of patients also. So, you know, we assume it exists somewhere. It just, some things may be a little harder to get than others.
Right. So another question pertains to how we measure, or how you measure success with data curation efforts, especially since, as you noted, it's not like there's a magic gold standard out there, some sort of external reference that tells us when we know we've achieved high quality, or, you know, that we've achieved the right answer. Can you sort of talk a little bit about how you think about that?
Yeah, I mean, it's a great question, and it's probably not it's something that we probably should have a better answer for. So there's certainly, you know, whether there's success in the sense that for the required checks and the required things that people need to pass for any given refresh or curation cycle, you know, were they able to clear those, right? So that's one measure of success, in the sense that everybody, you know, passed data curation this past cycle. Now, as people have noted, some of these things, you know, some of these bars seem a little bit low. And so, you know, what I would say is, you know, are we able to, for instance, turn some of these required checks, or these investigative checks that we have, into required checks? And you know, so we've sort of moved some of the things from checks where we're trying to understand what's going on and seeing whether or not these things can be resolved, to saying, you know what, everybody should have to pass this. And so now we've expanded the number of things that we're looking at. So, like, that could be another level of another form of success. And then, you know, there's for things like labs and meds, again, that, you know, have been a big focus, because they're an important part of what we're trying to do, and they've really been not standardized at all. Are we seeing movement? You know, are we able to get more and more of the data mapped to the right kind of standard? And so, you know, there you can look at, have we increased the number of labs that we have sort of available to us, and then also the volume of labs. And so I think those are, you know, those are other measures of success that we can try to use. But it's a great question.
Yeah. So we have time for probably just a couple more questions. So let me read one to you that is in the Q&A. It's about the process for incorporating clinical context into these data quality checks. Our expectations about what's noise versus signal may vary based on the way that medicine is practiced within specific disease areas, or even in different settings. Would this type of context only be incorporated in a study-specific setting?
Yeah, that's a great question. I think that's primarily where it would be incorporated. I think it could, you know, in some sense, it does appear like we've seen in some of the pediatric versus adult. And I apologize, my dogs were quiet for 54 minutes of this presentation, and then they decided to go nuts. But it's, you know, in pediatric versus adults, where if we're looking at, again, the blood pressure example, that's probably not a great clinical context. But, you know, what I would say is the broad check, it may not necessarily apply, but if you were to start to subset that in, like when I talk about the population reports, for instance, that's where you could see some of the clinical context. So, you know, do you expect to see patients with a certain condition having a lab test or a medication or something like that? So that's where I would say it would be in kind of the next level down, which is not something that we've developed as robustly as we would like within PCORnet.
Great, thanks. So back to the labs, can you speak to whether the foundational checks handle issues related to results or units and standardize those across sites, or is that part of the specific check? So think about A1C values of 7, 7.0, you know, 7%. Where exactly are those standardized?
Yeah, so that happens. We have in the outlier checks where we have, I apologize for the dogs. We have the median values for the different lab results. Different values for the lab results, and so that's where we'll look at whether the median value for a data mart is sort of out of range. There's interquartile ranges and other things that we use from the expected median, and actually that's where we've seen in some of our data checks where, you know, somebody may report a value of 7, and then the median for someone else is like 0.7, and so there it's just you know, is the unit of measure off, because, you know, 0.07 with a unit of percent is not the same thing as 7 with a unit of percent. And so that's the way that we tend to handle it from the some of our outlier checks.
Great. So I think this will be our last question. I saved the money question for the last question. So if your dogs bark at this one, we'll know why. Can you talk a little bit about just the resources that are required to do this kind of data curation? Sort of what do we know about what that looks like at the health system level, at the partner level, and then what does it look like centrally?
Yeah, so at the partner level, I mean, I think most of them have roughly an FTE dedicated to the data mart administrator type of person that's doing the ETL process to load the data model and then investigate the data checks. I think at the coordinating center, you know, the team would say that they definitely need more people. They're hard workers. There's essentially like a stats programmer who does the bulk of the programming. There's a project lead that, you know, does a lot of the specification kind of work. And then there may be some informaticist types that do deep dive analyses, and then like 1 or 2 others that help, you know, that help look at the results that come into the coordinating center when we're doing refreshes. So it's, I think given the scale, it's actually a relatively small team. And so I think, you know, what I would say is that can be expanded or contracted, I think, depending on like how many investigational analyses do you want to do in addition to just sort of running the system. And so, you know, I think we've had times at PCORnet where we're more of just keeping the lights on, just sort of keeping things going. And so the resources scale down. And then if we need to start doing, again, deeper dives or thinking about what's next, we may be able to add some additional FTE or additional bodies to the project.
Great. Well, Keith, I want to thank you again for presenting today's webinar. And I'd be remiss if I didn't say that in the chat, we did have some people who really liked having the dogs as part of this. So that was a bonus for many of us. As a reminder, this session has been recorded and will be made available through the Sentinel webpage. You can also find details and registration links for future Innovation Center webinars there. Again, that website is sentinelinitiative.org. This concludes our session for today. Again, thanks to Keith for a great presentation, and we hope to see all of you again next time. Have a good day.
Great, thank you.